Welcome to the first video episode of Biotech Talks, an initiative by Department of Biotechnology of Haldia Institute of Technology, where we will present to you about the modern works in the field of biotechnology. Today's topic is clinical approach towards the 21st century pandemic COVID-19. First, we will have a quick glance through the contents. Coming to the introduction. Severe acute respiratory syndrome coronavirus 2 SARS-CoV-2 is the causative agent of COVID-19. The first case been reported in Wuhan, China in November 2019. On March 11, COVID-19 has been declared a pandemic by WHO. Tests being performed are antibody test, reverse transcription polymerase chain reaction test, isothermal nucleic acid amplification test, etc. No licensed vaccines has been found till date. Many biotech firms are being undergoing researches from March 2020 to come up with effective vaccines. Now let's see the history of SARS. SARS-CoV-2 isn't the first SARS. Human race has already experienced SARS earlier. In the beginning of 21st century, total patients affected with severe acute respiratory syndrome coronavirus 1 SARS-CoV-1 is 8096 with 774 deaths with fatality rate of 9.60% and Middle East Respiratory Syndrome MARS is 2519 with 866 deaths with a fatality rate of 34.4%. SARS and MARS virus was introduced to the human species by palm civet and dromedary camel respectively. Assuming nCoV-19 to be a zoonotic virus, but its origin is still under research. Now coming to the outbreak of COVID-19. The spreading of coronavirus has immensely affected the whole world. Above is the world map showing the amount of affected in every 1 lakh. Then we can see the statistics which shows an exponential growth of COVID-19 towards a devastating result. As of 1808-2020, almost 20 million population of whole world is affected with 7,73,000 deaths. Now coming to the structure of COVID-19. The CoV particles are pleomorphic. That means it doesn't have a definite structure with a helical nucleocapsid. The diameter is approximately of 125 nanometer with 30 kilobase plus sense RNA genome. Due to the spike present on the surface plays an important role and is variable in maximum portion. The upper part, which is the most variable part of the nCoV-19 genome, is a receptor binding domain. Lower part is fusion machinery and more conservative and helps in membrane fusion and nuclear capsid payload in host cell. The spikes make the virus deadly as it makes the spikes more stickier, leading to damaging of inner vital organ by more cytokine production. Since the shape of virus looks like a crown, therefore the name coronavirus is derived from Latin word corona meaning crown or wreath. As scientists suggest, the protein present on spike can be useful for vaccination against COVID-19. Next we come to replication and gene expression. The nCoV-19 was annotated to possess almost 14 open reading frames encoding 27 proteins. This means the eukaryotic translations are generally monocystronic in nature. This virus can encode 27 different messages from a single RNA, making it very dangerous. Reasons for such behavior is still under research. 5' to PP1AB are non-structural proteins and rest are structural accessory proteins. Approaching towards the treatments for COVID-19. Main theory of dealing with drugs are antiviral, adenosine-nucleotide analog inhibiting RNA synthesis, etc. The many drugs failed during phase 2 and 3 due to unknown toxic side effects. The main theories of vaccines are mRNA vaccine, adenovirus vector vaccine, Russian vaccine, etc. Briefing about mRNA vaccine. It is made by pre-programmed mRNA and the genes from spike protein. Once inserted, spike protein helps to invade the virus itself and the programmed mRNA enters the cell. Upon entering, it affects the translation mechanism and forces to production of faulty message, thus damaging the very core processes of cell surviving. Eventually, the cell kills itself because of the faulty messages. mRNA vaccine works in the most subtle level known till date and is making its way good. Then, adenovirus vector vaccine. This vaccine is currently being made by Oxford University. In this case, adenovirus CHAdOx1, originally a virus that infects chimpanzees, are used as vector to enter the spike protein. Upon entering, it is encountered by the immune system. The system remembers the structure of the spike protein. So when the original virus is encountered, the percentage of survival of the virus is lessened as the immune system was trained to destroy it. And then the Russian virus, Sputnik V, standing on the fundamental theory of Oxford vaccine. The Russian vaccine is named Sputnik V in reference to first artificial satellite Sputnik I, launched by USSR in 1957. The vaccine is prepared by Gemalaya Research Institute of Epidemiology and Microbiology. It uses adenovirus as vector, both shots contain modified genes from spike protein. This should be taken in 2 shots 21 days apart. The Russian researchers have claimed to pass phase 1 and 2 but without any adverse effects, though no detailed results is published. Lastly, discussing about treatments for COVID-19 in India, initially the breakthrough in drug manufacturing for nCoV-19 is catalyzed by invention of hydroxychloroquine, though it failed in phase 2 causing unknown heart toxicity. Talking about vaccines, Indian companies like Biocon Seagull are working simultaneously besides Oxford University to manufacture a vector vaccine by using genetically modified non-replicating measles virus as vector to insert with spike protein in human body. So if you liked the video, then do hit the like button and subscribe the channel and hit on the bell icon to get the further notifications. Thank you.